Today, I am taking up the topic nephron, structure, ultrafiltration, and formation of hypertonic urine. In our body, nitrogenous waste materials are formed due to metabolism of proteins and amino acids. In all the animals, nitrogenous waste products are formed and they are to be excreted. These nitrogenous waste products are mainly three types: ammonia, urea, and uric acid. Ammonia is highly toxic but highly soluble in water. So, the animals that are aquatic and have simple bodies excrete ammonia directly into the surrounding water. The animals excreting ammonia are known as ammonotelic animals. Example: protozoans, porphyrins, cnidarians, etc. Even bony fishes excrete ammonia. Then, the animals that can spare water for excretion convert ammonia into less toxic urea. So, excretion of urea, or the animals excreting urea, are known as ureotelic animals. But excretion of urea causes a lot of water wastage. Animals belonging to phylum Annelida, then class Amphibia and class Mammalia mainly excrete urea. The animals that cannot afford to lose water through excretion convert the urea into uric acid and excrete. The animals excreting uric acid are known as uricotelic animals. Example: insects, birds, reptiles, etc. Then, there are various types of excretory organs in animals. There are phlegm cells in platyhelminth animals. Phlegm cells are a type of protonephridia. Another type of protonephridia, namely solenocytes, are present in Amphiaxis, belonging to subphylum Cephalocardata. In annelids, we come across nephridia. In insects, there are Malpighian tubules. In crustaceans like crab, there are Hoxer glands or green glands. In the vertebrate animals, there are kidneys. So in all the vertebrate animals, kidneys are the only excretory organs. So in human beings, we have a pair of bean-shaped kidneys located in the abdominal cavity. They are also known as retroperitoneal organs because only their ventral surface is covered by the peritoneum. So the kidneys are known as retroperitoneal organs. In each kidney, there are about 1 million nephrons. Nephrons are the structural and functional units of excretion. These nephrons are again two types: cortical nephrons and juxtamedullary nephrons. Cortical nephrons are confined to the cortex. They have a short Henle's loop and vasa recta is either present or absent. The cortical nephrons are 85% of total number of nephrons. That means they are the largest number and they are mainly concerned with the formation of diluted urine. Coming to the juxtamedullary nephrons, these are only 15%. They have a very long Henle's loop. Vasa recta is also highly developed. They alone are capable of producing hypertonic urine. So, cortical nephrons produce diluted urine and juxtamedullary nephrons produce hypertonic urine. Then, coming to the nephron, there are three main parts: Malpighian body, renal tubule, and vasa recta. The Malpighian body consists of again two parts: glomerulus. Glomerulus is a network of blood capillaries, and the Bowman's capsule. Bowman's capsule is a double-walled cup-like structure. In this cup-like cavity of the Bowman's capsule lies the glomerulus. The glomerulus receives blood through afferent arteriole, this one, and it sends out blood through efferent arteriole. Spelling, you should be very careful. First letter is A, here the first letter is E. See the diameter of the afferent and efferent arterioles. The diameter of afferent arteriole is much bigger than that of efferent arteriole. As a result, hydrostatic pressure is generated here, and in the glomerulus, blood is filtered. This filtration is known as ultrafiltration or glomerular filtration. The filtration is due to hydrostatic pressure of the blood. The blood and the cavity of the Bowman's capsule are separated by 3 layers. The endothelium of the capillaries, the basement membrane, and the podocytes of the Bowman's capsule. So the inner wall of the Bowman's capsule is made of podocytes. That means they resemble the shape of our feet. That's why they got the name podocytes. Between the podocytes, there are openings that are slit-like. And through these slit-like openings, the glomerular filtrate enters the Bowman's capsule. Then coming to the renal tubule. It is very long and it has 4 parts. Proximal convoluted tubule, this one. This is Henle's loop. This is the distal convoluted tubule and in the collecting duct. Here, the Malpighian body, proximal convoluted tubule or PCT, distal convoluted tubule or DCT, all of them lie in the cortex. Whereas the Henle's loop and the collecting duct lie in the medulla of the kidney. That point you have to remember. Then, the Henle's loop, this one. It is a U-shaped structure and having 2 limbs. This is the descending limb and this is the ascending limb. The ascending limb has again 2 parts. It has a thin segment, lower thin segment and upper thick segment. They got physiological significance, of which I will explain later on. Then there is also vasa recta. Vasa recta is a capillary network present around the Henle's loop. Henle's loop. Here, the efferent arteriole, that is the vessel coming out of the glomerulus, produces a capillary network around the PCT and the DCT. This capillary network is known as peritubular network. Peritubular network. From this peritubular network arises the vasa recta, like this. Then, ultrafiltration. I will have told you, due to high hydrostatic pressure, ultrafiltration takes place in the glomerulus and the blood plasma with almost all its constituents except the proteins is filtered, and that filtrate is called glomerular filtrate. So glomerular filtrate is nothing but blood plasma minus proteins. So the glomerular filtrate contains many of the useful substances like glucose, amino acids, and also waste products like urea, uric acid, etc. So the glomerular filtrate that is entering the Bowman's capsule flows into the proximal convoluted tubule. The proximal convoluted tubule is made of simple cuboidal epithelium. These epithelial cells are provided with microvilli or brush borders. Then, from the glomerular filtrate, the entire glucose is absorbed in the PCT. small amounts of glucose is absorbed through passive transport, and the rest of the glucose is absorbed by active transport. So through passive and active transport, the entire glucose is absorbed in the proximal convoluted tubule. Similarly, amino acids and other useful substances are all absorbed into the blood from the proximal convoluted tubule. Then, in the renal tubule, we find two types of absorption: obligate absorption and facultative absorption. Obligate absorption means compulsory absorption. Facultative absorption means need-based absorption. So absorption of water and other substances taking place in the proximal convoluted tubule is known as, and also in the descending limb of Henle's loop, is known as obligate absorption. This absorption occurs always. Whereas absorption taking place in the distal convoluted tubule and collecting duct is known as facultative absorption. That means only when there is a need, then only water and other substances are absorbed in the distal convoluted tubule and collecting duct. Then, some substances are also released into the glomerular fluid. Glomerular fluid from the tissues or from the tissue fluid. That is known as tubular secretion. Tubular secretion means release of some of the substances into the glomerular filtrate. Now let us see. In the proximal convoluted tubule, water is absorbed, bicarbonates are absorbed, sodium chloride is absorbed, and nutrients like glucose, amino acids are also absorbed. Then, the tubular secretion, that means hydrogen ions, ammonia, and potassium ions are released into the tubule or into the glomerular filtrate. In this way, absorption and tubular secretion takes place in the proximal convoluted tubule. Then, this is the descending limb of Henle's loop. The descending limb of Henle's loop is highly permeable to water. So water will be coming out of the descending limb of Henle's loop. See the concentration of fluids here. This is the cortex, this is the medulla. In the cortex, the osmolarity of the fluid is only 300 milliosmoles. And as we move from the cortex to deeper medulla, the osmolarity of the medullary fluid gradually increases. This is very, very important. As we move from cortex to the inner medulla, the osmolarity gradually increases. See, here it is only 300, then 600, 900, 1200. By the time the fluid reaches the hairpin bend of Henle's loop, the osmolarity of the medullary fluid reaches 1200 milliosmoles. So correspondingly, the osmolarity of the glomerular filtrate also increases from 300 milliosmoles to 1200 milliosmoles by the time it reaches the hairpin bend of Henle's loop. How it happens? Due to outward movement of water from the glomerular filtrate. Because the surrounding fluid has higher osmolarity, water escapes from the glomerular filtrate to the outside. As a result, that means by loss of water, the concentration of glomerular filtration increases from 300 to 1200 milliosmoles by the time it reaches the hairpin bend of Henle's loop. Then, this is the ascending limb of Henle's loop. I have told you there are two parts, thin segment and thick segment. Thin segment lies in the inner medulla. This is inner medulla, this is outer medulla. In the inner medulla, this is the thin segment, and in the outer medulla, this is the thick segment. So here, the glomerular fluid, as it moves up, it must get diluted. And by the time it reaches the cortex, the osmolarity of the glomerular filtrate must be around 300 milliosmoles. Then how this is achieved? This is achieved by the absorption of sodium chloride into the medullary fluid from the glomerular filtrate. So from the sodium chloride present in the glomerular filtrate is absorbed into the medullary fluid by passive transport in the thin segment. So in the case of thin segment, from the glomerular filtrate, sodium chloride comes out through passive transport. Then, in the thick segment, sodium chloride escapes or removed from the glomerular filtrate through active transport, which involves expenditure of energy. So in this way, through passive and active transport of sodium chloride out of the glomerular filtrate, the glomerular filtrate gets diluted from 1200 millivolts to 300 millivolts by the time it reaches the cortex. Then, in the distal convoluted tubule, based upon the need, sodium chloride, water, and bicarbonate are absorbed from the glomerular filtrate. Then, potassium ions, hydrogen ions, and ammonia are released into the glomerular filtrate through tubular secretion. Then, this is the collecting duct. The glomerular filtrate now enters the collecting duct. In the collecting duct, again, as the glomerular filtrate passes through the collecting duct, it encounters increasing osmolarity of the medullary fluid. So the medullary fluid is 300. By the time it reaches this point, it is around 1200. So as the glomerular filtrate passes through collecting duct, again the concentration of the glomerular filtrate increases. And by the time the glomerular filtration reaches the tip of the medullary pyramid, its concentration increases to 1200 milliosmoles. Then how this concentration is increased, how this happens? This happens by absorption of water from the glomerular filtrate into the medullary fluid. Then secretion of potassium ions and hydrogen ions into the glomerular filtrate. So in this way, by the loss of water and by the absorption of some ions, the glomerular filtrate concentration increases from 300 to 1200 milliosmoles. Here one more point you have to remember. From the collecting duct, some urea also escapes into the medullary fluid. This urea enters the ascending limb of Henle's loop. This absorption of urea is to maintain the osmolarity of the medullary fluid. Let us not bother much about that. So in this way, various parts of the nephron participate in absorption and tubular secretion. Then, now let us study counter current multiplier mechanism. There is a mechanism known as counter current multiplier mechanism in the kidney. It is very important. That means movement of fluids is in opposite direction. You take the Henle's loop. This is the Henle's loop. This is the descending limb. This is the ascending limb. See the movement of fluid. In this limb, it is moving in this direction. In this limb, it is moving in the opposite direction. So counter current, the movement of the fluid or glomerular filtrate in the Henle's loop is in opposite directions. In both the limbs, it is opposite directions. Then you take the vasorector. This is the vasa recta. Vasa recta is closely applied or very closely associated with the Henle's loop. This is also very important. Again, take the vasa recta. In the vasa recta, blood in this limb, blood flows in this direction. In this limb, blood flows in this direction. That is, in the two limbs of vasa recta, again the blood flow is in opposite directions. Then you take the movement of fluids in these two. Ascending limb of Henle's loop and descending limb of vasa recta. Again, this is opposite. In this it is in this direction, in this one in this direction. Again, you take these two. In the descending limb of Henle's loop, the direction of flow is like this, and in the ascending limb of vasa recta, the fluid movement is like this. Why it is like this? It has great physiological significance. If this arrangement is not there, the medullary fluid or the concentration of the medullary fluid is disturbed, and it is not possible for the kidney to produce hypertonic urine. Now let us see why this arrangement is present in the kidney. I have told you the concentration of the glomerular filtrate passing through the descending limb gradually increases from 300 to 1200 milliosmoles by the time it reaches the hairpin bend of the Henle's loop. That means water is continuously coming out. If this water continues to move into the medullary fluid, what happens? The medullary fluid gets diluted and the concentration gradient is lost. Once the concentration gradient is lost, what happens? Hypertonic urine cannot be produced. So to avoid this, what happens is the water coming out of the descending limb of the Henle's loop enters the ascending limb of the vasa recta. Here I have to tell you one more point. Blood in the vasa recta flows in this direction. So just like the glomerular filtrate, the osmolarity of the blood also increases from here to here. So the osmolarity blood is only 300 milliosmoles here in this descending limb of vasa recta. And by the time it reaches this bend, the osmolarity of the blood increases to 1200 milliosmoles. Again, by the time blood reaches the cortex level, the osmolarity is reduced from 1200 milliosmoles to 300 milliosmoles. Then, how this happens in the case of blood? See, here, water is coming out from the descending limb of vasa recta. This water coming out of the descending limb of vasa recta enters the ascending limb of vasa recta. Sorry, the water coming out of the descending limb of the Henle's loop enters the ascending limb of the vasa recta. It means as water flows into the blood of ascending limb, the blood gets diluted. So, by absorbing this water, the blood flowing through the ascending limb of vasa recta gets diluted, and by the time it reaches cortical level, its osmolarity is reduced to 300 milliosmoles. Then, in this limb, the osmolarity of the blood increases from 300 milliosmoles to 1200 milliosmoles. How it happens? The sodium chloride coming out of the ascending limb of the Henle's loop enters the descending limb of vasa recta. So, because of the accumulation of sodium chloride in the blood osmolarity increases from 300 milliosmoles to 1200 milliosmoles in the descending limb of vasa recta. In this way, the osmolarity of the concentration gradient of medullary fluid is maintained, and also water and sodium chloride are sent back into the bloodstream. So this is known as counter current multiplier mechanism, or simply counter current mechanism. This arrangement is also seen between our arteries and veins. Then, Regulation of kidney function. How the kidney function is regulated. That means how the hypertonic or hypotonic urine is formed. This regulation occurs in three ways. First one, the three are hypothalamus, juxtaglomerular apparatus, and ANF, atrial and natriuretic factor are involved. So, in the regulation of kidney function, three factors are involved: hypothalamus, JGA, and ANF. Now let us take up first the hypothalamus. When the loss of body fluids, that is blood or tissue fluid, happens what is the result? The blood pressure comes down. So loss of blood or tissue fluids causes a fall in the blood pressure. So whenever there is a fall in the blood pressure, the osmoreceptors present in the body detect that change. So the osmoreceptors detect the fall in blood pressure and report the same to the hypothalamus. Hypothalamus is the floor of the diencephalon. And below the hypothalamus there is the pituitary gland. Pituitary gland has three lobes: anterior lobe, middle lobe, and posterior lobe or neural lobe. So the hypothalamus directs the neurohypophysis or posterior lobe of the pituitary to release a hormone known as ADH or antidiuretic hormone. Antidiuretic hormone. ADH is enemy of diluted urine. That means it helps in promotion of absorption of water in the kidney. So ADH acts upon the nephrons and helps in absorption of water into the blood. So when water is absorbed into the blood, what happens? Blood volume increases. Blood pressure increases and comes to normal level. In this way, by absorbing water from the urine in the kidney, ADH promotes or increases the blood pressure. ADH is also known as vasopressin. Vasopressin means compressing blood vessels. So, ADH is also known as vasopressin, and the action of vasopressin is on the arterioles. Vasopressin contracts the arterioles, and when the arterioles contract, the blood pressure increases. In this way also, the blood pressure increases by ADH. Then, juxtaglomerular apparatus. Juxtaglomerular apparatus are JGA. I have told you this is the afferent arteriole, this is the efferent arteriole. The afferent arteriole comes into contact with the distal convoluted tubule or DCT. Here I have drawn DCT and this is the afferent arteriole. When these two touch, or when the afferent arteriole comes into contact with the distal convoluted tubule, juxtaglomerular apparatus appears and formed. The cells of DCT closest to the or attached to the afferent arteriole becomes thick and form a patch-like structure. This patch of thickened cells is known as densa macula. So densa macula is formed in the distal convoluted tubule. In the case of afferent arteriole that comes into contact with the DCT, here also the cells of the arteriole multiply and they get thickened and form a structure known as juxtaglomerular cells, or they produce juxtaglomerular cells. The juxtaglomerular cells and the macula densa together, they form the juxtaglomerular apparatus or JGA. So these together form the juxtaglomerular apparatus or JGA. So whenever there is a fall in the blood pressure, this JGA releases one enzyme known as renin. The spelling R-E-N-I-N. If you write R-E-N-N-I-N, it is different. R-E-N-N-I-N is formed in the digestive system. This renin, R-E-N-I-N, is formed in the kidney. So, whenever there is a fall in the blood pressure, JGA releases the enzyme renin. In the blood, there is one substance known as angiotensinogen. In the blood, there is angiotensinogen. This renin acts upon the angiotensinogen and converts the angiotensinogen into angiotensin 1. Then, angiotensin 1 is further converted into angiotensin 2 by another enzyme known as angiotensin converting enzyme. This angiotensin converting enzyme is present in the lungs. So, angiotensinogen is formed. Angiotensinogen 2. This angiotensinogen 2 increases blood pressure by constricting the arterioles. So, the name itself indicates angio means blood vessel. Tension means increasing the tension. So, angiotensinogen 2 helps in contracting the arterioles mainly, or the blood vessels, and as a result, the blood pressure increases. So, whenever the blood pressure falls, this JGA system is activated and blood vessels are contracted and the blood pressure is increased. This system is known as renin angiotensin system or RAS system. RAS means renin angiotensin system. Then, angiotensinogen has another function also. It also stimulates the adrenal cortex. In the adrenal gland is present just above the kidney. It has again two parts, outer cortex, inner medulla. So adrenal cortex releases one hormone, one steroid hormone known as aldosterone. So angiotensinogen 2 stimulates the adrenal cortex to release aldosterone, which promotes absorption of sodium and water into the blood in the DCT and collecting duct. I have told you the absorption of water and salts here and here is only not obligatory but facultative. So whenever the blood pressure falls down, this angiotensinogen acts upon the distal convoluted tubule and collecting duct and helps in the absorption of water and salts from the glomerular filtrate. In this way, the blood volume increases and it comes to normal level. There is also a third method called ANF mechanism. The upper chambers of the heart are known as atria. Atria collect the blood from various parts of the body, including the lungs. So whenever the blood pressure or blood volume increases in the atria or atrial chambers of the heart, what happens? They release this factor, atrial natriuretic factor. So atria release atrial natriuretic factor or ANF. So here the problem is different. In the first two cases, the blood pressure is decreased, but here the blood pressure increases. So ANF has to decrease the blood pressure. How it decreases? By dilating the blood vessels. ANF dilates the blood vessels. As a result, the blood pressure comes down. It also promotes excretion of water and salts from the blood. So by these two activities or actions, ANF brings down or lowers the blood pressure to normal level. In this way, these two systems and this system work in opposite direction and regulate the blood pressure and blood volume in our bodies. Though 1/5 of the blood coming out of the heart is supplied to the kidneys and many thousands of liters of glomerular filtrate is formed. 99% of the glomerular filtrate is absorbed back in the nephrons and only 1% of glomerular filtrate comes out in the form of urine. Per day, we produce only 1.5 liters of hypertonic urine. So this is about the structure, ultrafiltration and formation of hypertonic urine in human beings. Thank you.